Rock and roll.
Okay, now the question is, how do we make ATP? Well, what we have here right now is a ton of H plus stored up in the intermembrane space. It's all over the place. As a matter of fact, the concentration of H plus in the intermembrane space is about 80-fold higher than it is in the matrix. And that generates a pH difference. It's about 5.5 or so in here. That's fairly acidic. It's about as acidic as beer or Coke or what have you. And over here it's about 7.3. That tells you about 80% or 80-fold difference in hydrogen ion concentration. So that's a huge amount of H plus stored across the membrane. Okay, so of course the H plus wants to diffuse back. Problem is, it can't get through the membrane very well. Because, of course, H plus is charged, and the last place in the world a charged molecule or ion wants to be is inside a greasy hydrophobic membrane, right? So membranes are good insulators against ions getting across. You have to have special transport mechanisms to allow that. Okay, so now we have all this. The other thing is this. We've been pumping positively charged ions across the membrane, but we haven't taken any negatively charged ions across. So what's happening is now we're getting an electrical difference here across this membrane. Remember, we mentioned about the sodium-potassium ATP has generated electrical difference across the membrane because it took 3 positively charged ions, kicked them out, and took only 2 positive charges back in. Here we're pumping nothing but positive charges. So we get a voltage across the membrane and that voltage is about 180 millivolts. That doesn't sound like much, 0.2 of a volt, right? Your flashlight battery's 1.5 volts. But that's across that very narrow 4 nanometer wide membrane. The electric field is about 300,000 volts per centimeter. That's 10 times what it would take for a spark to jump across an air gap. So we got a really good electrical insulator. So we're storing electrical charge across the membrane. And that voltage difference across this membrane, across this inner membrane, is about twice what you find in ordinary cells. Okay, so we got a huge amount, we've got concentration difference and an electrical difference across that membrane. Now, does anybody know of a very, in fact, all you guys with these iPhones and all this stuff, and iPads and what have you, there's plenty of them inside all our electronics. These little electronic devices, what they do is they store electrical charge across an insulating barrier, and then you discharge that at will. Anybody know what those are called? Capacitors. So, capacitors do that. Now think of this. Hopefully nobody's gone through this experience, but ever been zapped by a police taser or something like that? I don't know if this happens, right? You have that here? Okay. But anyway, if you've ever been zapped by something like that, those things are powered by a couple of flashlight batteries, 1.5 volts each, 2, maybe 4. Yeah, and what it is, they got a really big, huge capacitor in there. They store up enough charge that when they fire it into you, they give you about 50,000 volts, granted fairly low amperage, but it's enough to bring up even a football player down, most of the time at least. Okay, and if you ever had a camera photo flash, on almost all kinds of cameras, even on some of these smartphones, to fire off that flash tube takes 30,000 volts. Yet it's being powered up by pen light batteries or your cell phone battery, whatever the case is. The reason that works is you have a capacitor, a really big one. It builds up a lot of charge and then suddenly discharges it with enough charge is released to fire the photo flash off, to knock a person on the ground if they get hit with a taser, whatever the case is. Now we're doing the same thing here, folks. This is a biological capacitor. All this H plus up here is storing up energy that if we let that loose, if we let it go back across the membrane into the matrix, that's exergonic, that releases energy. And folks, that is exactly what we're going to do. Because inside the inner membrane of the mitochondria is what I call the magic mushroom. You can tell where I came from, right? I look like it, right? Yeah. Oberlin College in the 70s. Okay, well I call it the magic mushroom because it actually looks like a mushroom. Because the stalk is embedded in the membrane, it's full of hydrophobic proteins, and then the cap is around it. The actual name of this enzyme tells you what it does. It's called ATP synthetase. And what it does is it synthesizes ATP. It takes ADP, phosphate, joins the two together to make ATP. Now, of course, that condensation reaction is very endergonic. So you have to have an energy source to do it. And what is that energy source? It turns out our magic mushroom has an H+ channel running through it. So now what happens is the H+ goes through this channel back into the matrix. That is facilitated diffusion. And that's exergonic. When that happens, the energy of that facilitated diffusion powers the production of ADP. So what oxidative metabolism essentially does is we use oxidation reduction reactions to actively transport H+ across a membrane, effectively to charge up a biological capacitor. And then we discharge a capacitor through our ATP synthetase. And that enzyme makes the vast majority of our ATP. Once we make it, then transport proteins take the stuff out of the mitochondria across the membrane. Of course, it takes whatever it needs, and then pumps it into the cytoplasm and keeps you nice and happy. Now, there was a name for this process. This process and this mechanism was very controversial at first when it was first proposed. You look back in 1970s, people didn't know too much about this. They knew the electron transport chain. They knew it took electrons from coenzymes and passed them oxygen. They knew about that. They thought originally that somehow the electron transport chain enzymes themselves use the energy of the oxidation reactions to directly make ATP. But nobody could ever find any evidence for that. So along comes a Scottish biochemist named Peter Mitchell. Mitchell had a different idea. He thought that maybe those electron transport chain enzymes actively transport H+, and then it got discharged through some kind of enzyme that made ATP, that used the energy of facilitated diffusion to make ATP. When he first proposed that, it's kind of an alternate, people start laughing at him. In fact, so did the granting agencies. This guy had trouble getting grants because his idea was so weird. Sounds horribly inefficient, right? Pump H+ across a membrane, charge up a biological capacitor, and discharge it to make ATP. People thought the guy's out of something. Okay. So he even had trouble getting research grants. But fortunately for him, he was old Scottish royalty type, you know. He had a family, huge family estate, a family castle, and money up the wazoo. So the lack of grant money did not stop the guy. So what he did is funded a lot of his own research and literally had a basement laboratory in the basement of his castle, kind of like Dr. Frankenstein, but a lot nicer. Okay. He started doing some interesting and elegant experiments, and when he published that stuff, people started thinking, my God, this crazy Scotsman might actually be onto something. The idea was this. If his idea, and he called this the Chemieosmotic, and I'll summarize that in a minute, mechanism called hypothesis back then, but it's the way things work. We'll explain that. Well, let's explain that right now. What it means. Electron transfer, I just call ET. Okay, electron transport chain enzymes actively transport H+ across a membrane. Part 2 of this chemiosmotic mechanism is facilitated diffusion is ultimately of H plus is ultimately used to power the ATP production. So cells make ATP by actively transporting H plus, in effect charging a biological capacitor, and then using the energy to facilitate diffusion of that same H plus to provide the energy for ATP. Yes?
What is the word after H plus, please?
Powers. I know, I flunk handwriting all the time. Okay.
You said flowers?
Powers. Powers. Okay, well, that was his hypothesis. Sounds bizarre, but he had an idea that could test this out. Let's suppose here we have a huge amount of H plus across this membrane in the intermembrane space. Now, ordinarily this stuff leaks back through our ATP synthetase, through the magic mushroom. But what would happen if we punched a bunch of H plus specific channels in that membrane, where the H plus would leak back through that rather than the ATP synthetase? Well, what would happen is this. The electron transport chain would operate perfectly normally. You would see electrons removed from NADH and pass to oxygen. However, what you would not see is ATP production, because the minute you pump the H plus across the membrane, it leaks right back. So you can separate the electron transport chain operation from ATP production. Now, the old idea that the electron transport chain enzymes directly made the ATP themselves, if that was true, you could not separate the two apart. If the electron transport chain worked, ATP was made. But if you can separate the two, just by punching H plus holes in the membrane, that supports Mitchell's hypothesis. So sure enough, he did just that. Turns out there's this drug called dinitrophenol, or D&P. Phenol is quite hydrophobic, right? It makes H plus specific channels in cell membranes. So it gets in cell membranes, we have these H plus channels all over the place. The H plus leaks out as soon as it comes in, and there's very little to go through ATP synthetase. And the result, ATP production grinds to a halt. But the electron transport chain is working perfectly unaffected. It pumps the H plus across the membrane like it's supposed to, passes the electrons to oxygen like it's supposed to. What it doesn't do, you don't see ATP production, because the minute you pump the H plus across, it leaks right back. So Mitchell's experiments could separate the two, saying that the electron transport chain enzymes did not directly make ATP. What powered ATP production was that H plus stored between the two membranes. Okay, so these kinds of lines of evidence and others and stuff started bringing a lot of people on board. Pretty soon this chemiosmotic mechanism became generally accepted. Now it's pretty much, Okay. And many years later, after being laughed at and having a hard time getting grant money, guess what? He gets a ticket to Stockholm. He got the Nobel Prize for it. Of course, he died a few years afterwards. It was pretty up in years. But anyway, Mitchell got the Nobel Prize for this work. He stuck to his guns. He had an interesting idea and experiments that could show how you could settle that issue. Okay, so pretty cool stuff. Now, one or two other things. It turns out the old drug, dinitrophenol, or D&P, had an old use way back in the 1900s and stuff. It was used as a diet pill. Because if you took dinitrophenol, what would happen is all the calories, the food energy you had, you would get less ATP production. So food, in effect, would not have the energy content as it ordinarily would, because you make H plus, pump it across the membrane, the H plus leaks right back. Now, what happens when the H plus leaks back? That energy has to go somewhere. What happens is it's used to generate heat. Now, the problem with DNP was twofold. One of them is long-term use caused things like eye damage and other kinds of damage. So when the newly formed FDA, one of the first things they did was take it off the market. It was the first of a long line of failed or dangerous dietines. Okay, but the other thing is the difference between a effective dose and a lethal dose was not all that great. So you take too much DNP and what happens? You feel like you're suffocating from the inside out because you're no longer making ATP. Meanwhile, your body temperature skyrockets because the energy of that H plus linkage is going into heat. Your body temperature skyrockets, you sweat like crazy, you suffocate, you pass out, and then you die. Good reason they took that stuff off the market. Yeah, nasty way to go, I would suspect. Okay, but it was a great research tool. Now, another interesting thing, and this is really more organismally oriented, it turns out long before Mitchell started performing these nice, cool, elegant experiments, nature had been beating him to the punch. Because it turns out there are certain tissues in certain animals that have regulated H plus channels that can allow the H plus to link back by generating heat and bypassing the ATP synthetase. And that is a special kind of tissue known as brown fat. And it's called brown fat because it's fatty tissue, but it looks a reddish brown in color. Reason why? It's packed full of mitochondria. Mitochondria have all these blood red proteins in them. Now, brown fat is found right beneath the skin of various organisms. The ones you find that have brown fat tend to be little small mammals that live in cold environments and infant animals, especially ones that live in cold environments. In fact, even human infants have a little bit of that. It disappears after a few weeks and stuff. But even we have a little bit of that ourselves. So here it is. It's right beneath the skin. Now what happens with brown fat? Let's suppose you start, ordinarily these channels are closed. They're gated channels. In fact, they're ligand gated channels. Channels are closed. H plus cannot leak out. The brown fat tissue is packed full of mitochondria. So much mitochondria that makes far more ATP than it can ever hope to use. So you don't need the mitochondria so much for energy production. You got far too many. But what will happen? Let's suppose you get cold. Especially if your core body temperature starts dropping. That's really bad. If your core body temperature drops too low, you get cardiac arrest and organ failure, and you die. Bad thing, especially when you're a little infant, little fuzzy infant mammal living up in the polar Arctic regions, like snowshoe hares, voles, lemmings, things like that. Okay, now, you get cold, and what happens? Hormones are produced, they go through the bloodstream, they get into cells, and then they cause these channels to open up. So, H plus starts leaking back. That means heat is released. And since the brown fat is right beneath the skin, it means it's a little heating blanket that surrounds your core body. It keeps your core body nice and warm when it's 50 degrees below zero and 40-mile-an-hour winds outside. That's what you often get up above the Arctic Circle in wintertime, right? So it helps keep these little critters warm. And like I say, even human infants have a little bit of that. And then when the core body temperature gets warm enough, what happens is now the hormones are not produced, the gates close, H plus does not leak back, and then you stay that way, and then once you get cold, you open them up again. So you can open and close these channels to generate heat. And the reason the brown fat has so much mitochondria is they're out there to produce lots of heat. They don't need anywhere near as many mitochondria as they have just to make enough ATP to keep them going. So you have all this excess mitochondria that can be used to generate heat when you need to do so. So that's a cool little physiological adaptation of this that some organisms have. Okay, one or two last things. Now this chemieosmotic mechanism is used in oxidative metabolism, but it is also used in the chloroplast and in photosynthetic bacteria by pretty much the same way in photosynthesis. They have enzymes that actively transport H plus across membranes, They have their own versions of ATP synthesis, which are slightly different than the mitochondrial versions, but they're there. So this is a very broad-based mechanism. Here again, this first developed in photosynthesis, and type of photosynthesis we're used to, that developed at least 2.5 and possibly as far back as 3.5 billion years ago, very far back in the early history of life. There's a big debate about how far back that happened, and contradictory evidence and stuff like that. But we can say at least 2.5 billion years ago, possibly further back. So this is a very ancient mechanism. Now, one or two last little points here. How does this guy work? It's actually very neat. In effect, oh, it's cool. It's really cool.
Is this brown fat?
No, this is the ATP synthesis. How does the ATP synthesis work? Okay, it sounds like a crazy mechanism, right? Go through all that trouble pumping H plus across the membrane, and then letting it leak back and making ATP. It turns out the ATP synthesis enzyme, which has about 20 separate subunits, is effectively a little electric motor. Because when the H plus leaks through here, what happens is the cap starts, the part of the stalk starts spinning around at about 60 times a second. And that ultimately makes the ATP production, because what happens, I'm just going to put one of them, there's actually 3 of these sites. It turns out there's 3 binding sites for ATP and phosphate. I have to enlarge this. Okay. Okay, so ATP and phosphate go in, and it turns out when they fit into that active site, and there's 3 of them here in this enzyme, it actually gets joined together to make ATP. The problem is, once ATP is made, it's stuck in the active site and cannot get out. So the enzyme can make the ATP, but it can't release it. That doesn't do any good. I mean, why make ATP if you can't let it out and let it do something useful? But the way this thing works is the stalk, when it spins around, there's a loop of protein here. There's a loop of amino acids. As it spins around, every time it's spinning, let's suppose this happens to be our binding site for ATP. Out in the stalk here, and this is this loop of amino acids. I spin around. Bang! Sorry. Okay, well, I just dislodged the ATP. I spin around. Part of it actually hits the binding site and pushes the ATP off. And then I spin around again. And I keep on doing that. And that's how we make ATP. Now, it sounds crazy, right? How efficient is that? Without going into gory detail, somebody did some elegant experiment. They found out how much energy does it take to make an ATP from ADP and phosphate. We know that. How much energy is released by the facilitated diffusion of like 3H plus ions, which is what it takes to make an ATP. We know that too. How much of that energy is actually used, that facilitated diffusion is actually used to make ATP? This rather crazy magic mushroom here. Mm-hmm. Well, if you take a typical electric motor, most electric motors, only about 20, 30, maybe up to 40% of the energy from the electricity is going into moving the electric motor around. The rest of it gets wasted as heat. That's why electric motors get hot when they run.
They get wasted.
Now, so your typical electric motors, 20 to 40% efficient. How efficient is this guy? 95% of the maximum the laws of physics allow. You have to lose a little energy. You can't do anything about it. That's fundamental laws of physics. Okay, 95% of the maximum possible efficiency. In other words, this little electric motor powered by a minuscule current of H plus ions is almost perfect. Far better than anything our technology has, yet it's only a millionth of an inch across. That's pretty awesome. But on the other hand, nature has had 3 billion years of evolutionary trial and error to optimize that. If you have a more efficient ATP synthesis, you make more ATP for each organic molecule, or for each set of electrons from the coenzymes. So the more and better you get, and now we've gotten to the point where this thing is the height of perfection. There's almost no more room for improvement. The laws of physics prohibit it. So that's why I call this marvelous little molecular machine, that's why I call it the magic mushroom, because its operation is truly magical, and it's far better than anything our technology has to offer. So with that, does anybody have any questions here? Because if not, I wish all you folks a good Thanksgiving. Hopefully I'll see you guys on Tuesday.